Okay, well, I think we might as well get started here, and we have a number of participants joining, and anyone else who joins. The webinar will be recorded and available on our website following uh, today's talk. But just to start off, thank you all for coming. My name is Ward Detweiler. I'm the president and CEO of SpinTech, and I'm joined by our founder and chief science officer, Dr. Mark Hakey. And what we'd like to do today is to introduce you to our stage technology, really kind of framed around what I think is a very important topic, and I think all of you will likely agree, which is how the radiology world is going to respond to the kind of post-COVID imaging surge that is on the horizon once we see kind of the normal return back to imaging, and we have to deal with all these patients who've been gone without kind of the routine imaging they've needed over the past several months, and also talk about how stage we think is really an ideal solution for addressing that issue. Let's get back in here. So just to start off, I'd like to give a brief overview of our company and introduce ourselves. We are a fairly young company, just started in 2017, but based on decades of research and innovation by Dr. Hakey and his team at MR Innovations. We are working with over 50 global research sites to validate these technologies and establish new clinical use cases. We've developed a very strong IP portfolio, used our technologies to publish over 100 papers with 10,000 plus research references, processed over 8,000 cases as well, and have our first product, which is Spin SWI, cleared through the FDA, and now we're bringing stage forward as well. Just at a glance, what we're going to be introducing you to today is our stage software platform, which provides quantitative multi-contrast and standardized MR imaging, reducing the time required for comprehensive brain MRI by over 40%. On top of that, we also have a suite of AI-driven automatic detection and quantification tools, really providing rapid insights to the radiologist at the point of care, and also providing a really strong clinical and economic value for practices. So much, sorry. So as we look at the problem that we're all facing right now, and we don't really need to tell you any of this, COVID-19 has caused just a really an incredible disruption to radiology over the past months. We've seen upwards over 50% reduction in radiology exams during this period, but that demand has not disappeared. What that means is now we have millions of unscanned patients that we're going to need to see, that will need to be absorbed by radiology groups in the future, on top of normal demand and on top of some new pressures. And what this means is that as imaging volume comes back, really radiology groups really need to be prepared for this new environment and for this new world. Now, as we look at some of the recommendations coming out from groups like RSNA and others, there are some suggestions in terms of how we can help address this challenge. Things like increasing time between patients to add safety and sanitization protocols, using abbreviated or accelerated MRI protocols to help shorten scan times, and also driving extended scanning hours to help address this new demand. But all of this has an impact on radiology as well. It means tighter workflows and strained staff, the potential for missed diagnoses because of maybe you're not collecting all of the data you might normally collect for these patients, and then also a need to recoup financial losses and also find a way to remain profitable and successful as we move forward. And that's really weird, sorry. What we think here is with Stage, we've really come up with what we think is a really ideal solution to addressing this challenge. Stage stands for Strategically Acquired Gradient Echo, and that's really where it all starts. We use a proprietary rapid gradient echo sequence that's acquired in 5 minutes. This can be set up on any manufacturer using already clinically approved sequences that we just have edited parameters for. After this data is collected, it is transferred directly to an integrated processing hub that automatically processes this data and returns 15 enhanced multi-contrast quantitative outputs right back to the PACS, right where the radiologist is already working and where they're already looking. So no need to open up a new interface or use any new tools. We're really trying to minimize as much of the workflow disruption as we can. On top of this, we also have a suite of automatic detection tools that can use this data to provide automatic biomarker detection or quantification within the brain to help drive better diagnoses across a range of different diagnostic areas. Now, we don't collect everything with Stage, so we recommend for our kind of comprehensive brain protocol adding a T2 flare and a DWI on top. And what that means is that at 3T, you're getting a really comprehensive brain protocol in just over 9 minutes That's a considerable time savings compared to conventional scanning. That's leads to increased patient throughput, less patient time in the scanner, and also improved ROI for our customers. Um, Now, what this means ultimately, though, is that really you're no longer thinking about what am I going to run, just what am I going to look at, because all of the information that you are going to want is going to be there automatically on every single scan. And we've used this technology to image patients across a whole range of different diagnostic areas, including stroke, traumatic brain injury, Parkinson's, MS, and dementia, and have automated tools for biomarker detection across a range of these conditions as well. Now, as we look at the value back to the radiology group, we think we have a very strong proposition here, because 40% faster brain scanning means that you can see conservatively probably another 10 to 15 patients each week, leading to over $500,000 of additional reimbursement every year just from a simple software add-on. No upgrades to equipment, no purchasing a new MR system. So we think that is a really strong financial incentive, but also on the workflow and diagnostic side as well, through improved detection and improved patient outcomes, improved clinical workflow, data standardization across different machines, which Dr. Hakey will talk a little bit once we get into the more technical side of the talk. And then also increasingly important, and maybe most important, is improved patient experience, because nobody likes to be in that scanner for a minute longer than they have to. And it's increasingly becoming an important metric for determining reimbursement, at least here in the United States. I know we have some international attendees here as well. Now, on top of that, we also see this as a really important platform for the development of future AI tools. As you're probably aware, developing and deploying AI at scale on MR data can be incredibly difficult because some of the inherent challenges with this data. MR data, as compared to CT data, is not standardized. It's often not quantitative. It can take a long time to collect. And all of these are really roadblocks to developing new tools and also deploying them across multiple sites. And that's why I think we've seen most of these tools really living in the research world in individual sites up to this point. But we've really solved this challenge with Stage by producing standardized quantitative data that is collected incredibly quickly and is consistent scan to scan, meaning you essentially have a big data protocol every single time. And while we've already started developing our own tools, and this is an example of our first AI tool, which is used for the automatic detection of cerebral microbleeds, which is performing very well in terms of sensitivity and specificity, and performing all but some of the best human readers who happen to be on our team. We also know that we're never going to develop everything. There are great ideas all over the world, and hopefully in some of your institutions, and maybe we can work together on some of those. But it really provides a platform that we see for the deployment of some of these AI tools as we go forward. And we're already validating that with a number of third parties. So on top of this data, you can still run the normal quantitative reports that you might be used to. Here's an example in working with Qmenta and running some of their volumetric reports on top of this data. So what you're getting is really clean, very tight data that actually can improve some of these reports as well. So before we get into Dr. Hakey's talk, who will start taking us through some of the more technical aspects and some of the clinical use cases of Stage, just wanted to talk really briefly about how you get started using this. And really the first step is for us just to talk, to sort of understand your needs, understand what systems you're running, so that we can really tailor a solution that's best to your site. With that, we then will develop the sequence specifically for your machines, and they're very easily loaded onto the machine. We can either use the existing sequences that are on the machines and just edit parameters and save the Stage sequence, or depending on the system, we have exam cards or EDX files that we can load. So it's really quick and easy to get the sequence up and running on the machine. And then from there, we'd have you run a few little test scans, show you the data, show it in action, and then move to installing the Stage box, which is just a little medical grade mini PC. It's a very light footprint that sits on your network. It takes probably under an hour to get this installed and trained. And then from there, you're off to the races and are able to go and start collecting data and processing right at your own site. So that's really the high level of what we're working on. And I'd like to go turn it over to Dr. Hakey. Also, as we're going about this, if we have any questions that pop up, feel free to drop them into the Q&A box, and we'll address those questions either as we go or at the end, and then maybe open up for a little discussion at the end. So with that, I'm going to stop my portion here, and we'll turn it over to Dr. Hickey.
Thank you very much, Ward, for the introduction. And let me go to my slides here. So Ward is giving you an overview of the potential of using some of our software and sequences. I would like to particularly today talk about a comprehensive imaging protocol for neurological sequelae in COVID-19 patients using some of the technology that he's introduced and other aspects of MRI. So the COVID-19 pandemic has affected now over 10 million people with a worldwide fatality rate of about 5%. One of the key questions now with clinical care in COVID-19 is, what are the long-term ramifications of the vascular problems associated with COVID-19 infection, leading to neurological deficits in those patients who have recovered? COVID-19 appears to have strong effects on the vascular system through the cytokine storm effect, where the endothelium is compromised, causing ischemic injury and stroke from thromboembolic phenomena and tissue damage from extravasated hemorrhagic products. Some estimates today suggest that roughly 1/3 of the serious COVID-19 patients are afflicted by vascular-related neurological sequelae. A recent paper in the New England Journal of Medicine by Helms et al. Has noted that a majority of patients had intracerebral hemorrhagic lesions with a more severe clinical presentation. So neurological manifestations generally can include ischemic or hemorrhagic cerebral vascular diseases, cranial peripheral neuropathy, new onset or seizure or worsening of a previous diagnosis of epilepsy, new onset or worsening of a previous diagnosis of multiple sclerosis, impaired consciousness, new cognitive deficits or worsening of previous cognitive deficits, as well as neuromuscular diseases including myalgias, encephalomyelitis, myelopathy, sleep disturbances, headaches, ageusia, anosmia, ataxia, and dizziness. MRI offers a variety of imaging approaches to be able to image these effects. FLAIR is a technique to detect inflammation in white matter lesions. STAGE, as Ward has noted, can give us up to 15 different types of image contrasts and quantitative information, including mapping water content and T1 changes, as well as susceptibility mapping. MR angiography and what we call stage MR angiography, which I'll show you a little bit later. Quantitative susceptibility mapping, perfusion-weighted imaging, and diffusion-weighted imaging all offer us important information for studying neurovascular disease. So STAGE offers us 8 qualitative and 7 quantitative pieces of information in 5 minutes with a good resolution and full brain coverage, including T2, FLAIR, and DWI, less than 10 minutes. STAGE is becoming quite well published in the last 3 years. There are many references to this. So let me tell you a little about how STAGE works. So in STAGE, we collect 2 SWI-like sequences, or 2 3D gradient echo sequences, one with a low flip angle of 6 degrees and one with a high flip angle of 24 degrees. The low flip angle gives us spin density contrast in the gray-white matter, and the high flip angle gives us T1 weighted contrast for white matter, gray matter, and CSF. Now, if we combine these 2 images, we can create what we call a T1 weighted enhanced image from the STAGE processing. And here you can see that the basal ganglia are better shown, the putamen is better shown. It's just a much nicer contrast than conventional T1 weighted image. We can also produce the quantitative T1 maps and spin density maps, and you can see that the T1 map looks almost identical to the T2 weighted image. And that's because water drives T1 and T2. If you have a lot of water, T1 will be longer, T2 will be longer. And hence it's no surprise the T1 map looks just like T2. So if we combine all of these, here's 10 example images of this. The original T1, our enhanced T1, the spin density weighted, the T1 map, and the water content. The SWI and what we call true SWI that removes geometry dependence from SWI, a poor man's MRA, an R2 star map, and QSM for quantifying iron content. So all of this comes out of in 5 minutes. And this is just one example from a 3T data set. We can add to that DWI and ADC maps and either use a T2 flare, a 2D T2 flare or a 3D T2 flare to get good contrast for white matter lesions. So some examples of this, here's an example of a case with hypertension that has bleeding in the basal ganglia and the thalamus. This is the SWI, the central image is the QSM showing the susceptibility or the iron content in each of the lesions. And the right-hand image is our new stage MRAV, showing that we have eliminated the background from the brain here to highlight just the vasculature. We're using this a lot in stroke at the moment, and at Tianjin First Central Hospital in China, they've done several hundred cases of this. Here's an example of the double echo version of this. You can see the poor man's MRA is actually pretty good in this case because it's a higher resolution case. You can see a blockage here of the right middle cerebral artery. You can see the thrombus on the short echo SWI, and you can see the asymmetrically prominent veins on the long echo SWI, which is an indication of perfusion deficit, so slower flow, increased levels of deoxyhemoglobin. And this is the QSM image showing both the veins and the thrombus. Just to give you an idea of what you can get from SWI and QSM, this is an example of an SWI image after someone had 2 cups of coffee. This is the image before caffeine in the middle and after caffeine on the right-hand side. And here you can actually measure changes in oxygen saturation. Because caffeine is a vasoconstrictor, it slows the blood flow, causing an increase in deoxyhemoglobin levels. And so after caffeine, the oxygen level drops from 70% down to 60%. Here's an example in stroke showing the perfusion deficit using MTT. You can see the highlighted SWI effect here. And after treatment, the perfusion deficit goes away and SWI clears up. So asymmetrically prominent veins can be a marker for tissue that is still functioning and using oxygen. Here's a more dramatic example of that with a complete hemibrain effect. You can see the asymmetrically prominent cortical veins. On one side, you can see the increase in deoxyhemoglobin, and on the far right, you have the perfusion image that shows a deficit of blood flow here. And then on the left image, you have the diffusion weighted scan that shows there's a big mismatch between the change in flow versus the damage that occurred on DWI. So stage MRAV is a new technique that we use to let us remove the background signal. And I want to show you an example here of some MRAV. Again, in this case, we have a blockage of the left middle cerebral artery. And we can look at the different parts of the brain here. So we move up to the top part of the brain. We can get QSM from this. And then we can overlay this data on the original magnitude images. So we can have an overlay of the arteries on top of this. We can have an overlay of the veins on top of this. And there's no misregistration artifact. Everything sits in the same domain. That's a big advantage of this technique. And this is an example of the type of images that you can get if you run this with high resolution. Shows a lot of the small vessels, making it very useful for studying stroke and probably COVID-19 as well. So speaking of COVID-19, this is a case from the Poyaji paper from Henry Ford Hospital. Here you can see that the SWI shows a significant bleeding in the thalamus at different levels and different locations. So being able to map out all of these vascular features in COVID-19 is going to be important. So in conclusion, imaging these patients should make it possible to correlate vascular damage with neurological outcomes, determine if microbleeds are more prevalent than thrombi, to ascertain what age groups suffer the most from this, to determine if there is a race or sex dependence, and determine which patients will best benefit from treatment with heparin, for example, if coagulopathy is the issue. In the future, we believe it will be critical to image and map vascular damage and microbleeds in recovered COVID-19 patients with neurological sequelae. MRI measures are likely to be correlated with functional outcomes relevant to the neurological deficits. So our goal should be to show that the vascular pathology is able to predict the severity of these clinical neurological dysfunctions. Thank you very much.
Great. Well, thank you, Dr. Ege, for bringing us through that. We have actually a few questions that have popped up here. The first is what field strengths can STAGE work with? And one of the great things about this, we've actually run this all the way from 0.35 all the way up to 7 Tesla with great results. Right now, though, in terms of the clinical version of STAGE that is going through the FDA right now, it's just going to be at 1.5 T and at 3 T. But again, it can work at any field strength and on any equipment. Another question that we had come up was what kinds of organizations are using STAGE right now? So as I mentioned, we have over 50 research sites around the globe who are using this. And mainly they're using it either in very specific, clinical domains. So we have certain sites that are very focused on stroke or Parkinson's, for example. They're continuing to publish papers and we're helping to support their research as they go forward, also establishing new clinical use cases. But on top of that, we also have a number of clinical sites in the US that are now using this as part of our trial. We have some major university systems using this and also some independent imaging centers, parts of larger chains. So we really think it's a great tool, not just for the research side, which is sort of where we were born, but also a very critically important tool, for clinical, for just clinical radiology as well, solving a number of these, just big pressing challenges that we have right now. Well, another question that came out says, can this run on any scanner? What if we have several scanners from different manufacturers? And that's a great and really important point, I think. So this does work, across manufacturers, and we've run this, I think, on pretty much every major manufacturer and a lot of sort of other smaller groups as well, and some international manufacturers. And this is actually a question that comes up in terms of a challenge from radiology groups that say, I'd love to try something new, but I've just got this challenge where I've got data from all these different machines and it doesn't match, and we can't review things, and we have to scan people at certain sites and keep them on the same scanner. And that's one of the really beautiful things about STAGE, is that it standardizes the data across machines as well. So your quantitative values on a GE system and a Siemens systems will be the same. And we validated this in some of our papers, and actually have shown that this can work. And we'd be happy to kind of show you this in real time at some of your sites, just to really show the work. Another question is, does this require a separate interface? So no, and that was very intentional. I know there's a lot of tools out there that are very neat and provide a lot of nice processing ability, but they require you to go and open up a whole new program and kind of break your workflow. And we designed this in such a way that all of our processed images are going automatically right back into the patient record, into the PACS. So it's almost like this is built right into your system, even though it's just a very simple add-on solution. And I think that's really important. We actually had one of our early researchers who was talking to this, who said, I love this. I'd use this on every brain injury case I have, but if I have to spin around in a chair and do 3 extra mouse clicks, I don't care if it cures cancer. I don't want to use it, because that's just, you guys all live this. This is the reality of how tight your workflows are. So we try to be very mindful of that as well. Let's see. I think that's all of our questions for now. I don't know if Mark, if you have any closing thoughts on this or any comments.
Well, I can understand that a lot of sites in the US and in China too, they do often have magnets from different manufacturers. So, it's important clinically to be able to, run this both at 1.5 T and 3 T. Now, one thing I should say, to be fair, is that, I can run this at 7 T with incredible results, partly because I can run it faster and I can run it with higher signal to noise at 7 T. However, with the new 3T systems with 64 channel head coils on Siemens and 32 channel head coils for some of the other manufacturers, the signal to noise on these 3Ts is kind of approaching where a 7T was 5 years ago. So we've designed Stage to have very good signal to noise in its standard clinical application. You can also run this at 1.5T, but instead of 5 minutes, it's going to take you 7 or 8 minutes, depending on the resolution that you're running. It's going to take a little longer because you have to run a slightly longer TRs associated with it. But it's still quite viable, and it's not, incredibly longer. But 1.5T also has slightly less signal to noise, of course. And one of the ways that we try to deal with that is to slightly change the thickness from 2 millimeters to 2.8 millimeters. And then you can run it actually much faster. So it just depends on exactly the application that the physician is going to use this for. It's very flexible, like any of your other scans. If you really want to do high resolution, for example, right now we're doing a big study on Parkinson's. We've done several hundred cases. We're using 0.67 by 0.67 by 1.34. That takes 5 minutes a scan. So it's a 10-minute scan, but it provides incredible clinical information about diagnosing Parkinson's compared to healthy controls. So it just depends on the application. But I think the standard rapid 3T 5 minutes is a very robust and powerful tool.
Well, great. Thanks, Mark. One of the other things that I want to add that we didn't really get into is, we've done this for so many different conditions, and we can go on and on. I think we've got a whole nother presentation that is in the hundreds of slides that details out a lot of those use cases. But you can find a lot of that information on our website. And also, we'd be very interested in talking with you and with your sites to learn some of your more specific research areas, because one of the things that actually helps advance this tool is learning from some of the radiologists that are using it to help even educate us on some of the new uses that they see, because it's almost like this is such a powerful tool that at the base version, it's, I think, very applicable and very valuable for sort of just normal clinical radiology, but there's so much capability with this that we can really solve a lot of really unique both research and clinical needs. So I'd really like to welcome you to reach out to us. You can get in touch with us through email or reach out through the website to set up a webinar, set up a demo for your group. And we'd love to talk more and learn how we can help implement Stage and hopefully help drive you into the future of neurological imaging at your sites. So with that, I'd just like to say thank you to everybody for your time, and I look forward to talking to all of you very soon. So thank you very much.
Thank you very much.
Right.